Great. Thank you very much. It's really nice to come and talk about some of the work we've been doing in Zimbabwe. And quite a few of you will have attended some of the talks that I have given, telling you about the exciting national control program we have started and implemented in Zimbabwe, and I've given a lot of the background that went into that program. But what I'm hoping to do today is to talk about human helminth immune responses and show you some of the really basic scientific work we do in the lab that has helped us communicate to policy makers and to shareholders. Because a lot of what we do when it comes out in the media or when it comes out at the human health level, all you ever hear are the policies and the implementation. And the politicians and the stakeholders do not tell you about the science. And what I'm hoping to do is convince you that any advice that we give to policymakers and to governments and stakeholders involved in national control programs is really grounded in solid laboratory scientific research which has a strong evidence base. So today I'm going to talk about human helminth infections. And I'll give you a little bit of an introduction of what those infections are. So helminths are very, very important. Here I'm just showing the various ways of exposure of the particular helminth I'm going to talk about. They are parasitic worms. They infect over 2 billion people worldwide. The one that I'm going to talk about a lot is Schistosome hematobium. So this is commonly known as bilharzia. It causes snail fever, and it is the most prevalent human schistosome species in Africa. It's a very old infection, so you have records of it in the Dead Sea Scrolls. But also in the Bible, if any of you have read the Bible, you'll have come across the curse of Jericho. There was a very nice scientific paper in the 1970s explaining that the curse of Jericho was actually bilharzia infection. So when the Israelites were coming out of Egypt, which basically is the focus of Schistosome hematobium infection, as they made their exodus, they were infected with schistosomes. When they got to the wells of Jericho, they peed all over the area because there were no toilets, and they were already the vector snails, which then picked up the infection and were very heavily infected, so that whenever everybody else came to settle in Jericho and used the wells for water, they got heavily infected, and the infections were very heavy and caused lots of people to get sick and to die, and that was the curse of Jericho. But that's not what I'm going to talk about today. Today I'm going to focus a lot on the natural immunity, and I've highlighted it here because it really is the crux of some of the work that I'm going to discuss here. Natural immunity to schistosomes develops slowly with age, but the important thing is it does develop, and it is protective against infection and pathology. The only method of control we have is treating people who are infected with the drug praziquantel, and I'll talk a little bit about that. And as of today, we still don't have a registered vaccine. So this is a very important control tool we have. When we go into an infected population, what do we see in terms of disease? Well, we see a range of disease and morbidity. The first one is hematuria, or blood in urine, and dysuria, which is pain at urination. Hematuria is very common, and it is usually an indicator of how heavily infected you are. And usually around the ages of 9 to 15, people carry the heaviest infections. So in some communities, actually, because hematuria is so widespread, it is taken as a sign of maturity, particularly amongst boys. Schistosomiasis is also a very important cancer agent. So Schistosome hematobium is classed by the WHO cancer group as a group 2 carcinogen. That means it has the possibility to cause cancer. Bladder cancer is the 4th most prevalent cancer in endemic areas. And in something like 90% of the male bladder cancers, they're all due to Schistosome hematobium infection. If we look on the female side, it's called urogenital schistosomiasis because it does cause genital schistosomiasis, where the eggs are deposited in the genital tract, and that causes a lot of scarring and fibrosis. That reduces fertility and also increases susceptibility to HIV and sexually transmitted diseases. So very, very important for reproductive health. Impaired memory and cognition, one of the less specific effects of urogenital schistosomiasis. So when young children are infected, they carry very heavy infections, and this affects their memory and cognition, and ultimately affects childhood development. The eggs produced by the parasite, which I'll talk about shortly, will be trapped in the liver and kidneys and also the spleen. And what you get is an immune response directed against the eggs and causes the spleen and the liver to enlarge. So what we have here is an enlarged spleen and liver. Now, if we stop for a minute and think, schistosomiasis occurs in the same areas where you have malaria. Malaria is also associated with pathology of the spleen and liver. So this poor chap will probably have been infected with Plasmodium and schistosomes, and they've exacerbated the pathology of each other and ended up with the hepatosplenomegaly. I will come back to that because that is a very important aspect we must think when we're implementing a control program. Pathology is progressive and it starts in young children. That's very important because that's what underlines the control programs and when and how it is targeted. And I will explain that in a minute. So I mentioned that the only control strategy we have is anti-helminthic treatment. Biltricide tablets, that's the common name, contain the drug praziquantel, and it is issued as white tablets that are that big. In a different talk, I shall tell you the problems that causes when you give it to young children, but not today. Those tablets are taken as a single dose, and that's very useful because you do not want infected people to come for repeated dose because you end up with less compliance. So what do we know about praziquantel? It is safe and highly efficacious. For using it with, on population interventions, this is a very important thing. It is very cheap. It costs about 50 US cents per dose. Now, actually, the tablets themselves will cost about 10 cents, but the 40 cents is the operational cost. So you have, if you're doing a national control program, you have to go out and give the tablets to people, and that's why it comes to about 50 cents a dose. It works synergistically with the immune system, and it kills adult worms and reverses pathology. So how are we controlling schistosomiasis at a global level? It was really nice to see sort of end of the 90s, beginning of the 2000s, a real global push for the control of schistosomiasis. And this has been aided by two developments. The first one was the Millennium Development Goals. So MDG6, the aim was to combat HIV, AIDS, malaria, and other diseases by 2015. Schistosomiasis is contained in the other diseases. Second one followed soon after. I think we got excited and got momentum, so we thought, well, let's go for it. The World Health Assembly Resolution 54.19 followed soon after, and this was to treat at least 75% of all school-age children at risk of schistosome morbidity by 2010. That's why I have this photograph, and if you go to my group webpage, you'll see this photograph. This is because these are the school children. They carry the heaviest levels of infection, so we target them for treatment, and that's why we're targeting 75% of these school-age children. As I say, we got very excited, and in 2012, the World Health Assembly issued another resolution, 65.19. They thought, we can do this. Why not go for elimination? So this one is elimination. For most of us, we're still struggling around here. And not all the countries that are affected by schistosomes have implemented the World Health Resolution 54.19. And at the moment, we are working a lot at this stage. So I'll focus on that today. Okay, so I mentioned at the beginning of this talk that we conduct a lot of our studies in Zimbabwe. So this is Zimbabwe. It is sub-Saharan Africa. It's a very nice country, but it also has schistosomes. So we decided to have a look at how much infection was around in Zimbabwe. So in 2010, our group here in Edinburgh and our colleagues in Zimbabwe, so somebody within the Ministry of Health and at the University of Zimbabwe, we mapped the distribution of schistosomiasis in Zimbabwe. So it's represented here by the little green dots and the red dots. The size of the red dots increases with the increase in schistosome prevalence. What you can see is basically this half of the country, there's a lot of schistosomiasis, and on that half there isn't. And if I gave you about half an hour to think why, you probably realize that the reason we have lower schistosomes that side is because they're very dry. And you require fresh water for transmission of schistosomiasis. So the statistics we came up with were basically we have a national prevalence of 35 5%, with a regional variation of 0 to 80%. But in a lot of places, about 100% of the 8 to 11-year-olds are infected. That's very heavy. We communicated this to the Minister of Health, and as I mentioned, this was the basis for the National Control Program. And this was all done in 2010, and actually the results in this map have only been published in the past month. So this is what we published from that survey. But the most important bit of the publication is this bit that says a national plan of action for the control and elimination of schistosomiasis. So we presented this to the Minister of Health in Zimbabwe, and advocated to have a national control program, and the journey we took from there to end up basically here is an entire talk on itself, so I won't dwell on that. The important thing is, in September, in fact I think it was the 10th of September, the government of Zimbabwe launched a mass drug administration against schistosomiasis. So here's all the fanfare and all the celebrations for launching the program. This was targeted at school children. And we have lots of photographs to show for it. So these are children queuing up for treatment. And we also included preschool children. And once you had got your treatment, it became such a status symbol. You got your T-shirt and you could wear it. I even have one of those. That's all said and done, and that is very good. The question we now ask is, what are the long and short-term consequences of mass drug administration programs? We've gone out, we've given millions of Zimbabwean children these tablets. We are going around the world saying, this is a good thing, everybody else join us, but what's our evidence that it's a good thing? So this is what I want to talk a little bit about that. So the question I ask is, what happens afterwards? Well, we're interested in the short-term effects. If you're a government minister and you're talking to your Minister of Finance, he's going to want to know the cost effectiveness of the program. Is it worth our while doing that? So we, as the scientists, have to find a way of determining whether the treatment is effective. From the person receiving the tablets, from their point of view, you're interested in the side effects. What is this going to do to me? I take these tablets, you've told me that they're safe, but given your sources of tablets or the way they've been transported or anything, are there going to be some adverse events? Long term, what are the effects on host morbidity? So are you actually reducing the amount of disease I carry? What about on my overall host health? If there are side effects, are they going to spill over into other conditions? What are the effects on transmission? We know that these parasites are passed from one person to another via a snail vector. So are we going to reduce transmission? And what are the effects on the parasite populations? We are subjecting the parasites to very heavy drug pressure. Are they going to evolve drug resistance? So these are all very important questions. This one here is a whole lot of another talk. I'm not going to discuss that much today, but I'm going to concentrate on the ones that are related to the host health and host response. So my research group here in Edinburgh we were mandated with the task of being the external monitors and evaluators of Zimbabwe's national control programs. So we're independent evaluators, and in our monitoring and evaluation of Zimbabwe's national control programs, we follow the basic World Health Organization guidelines on how you monitor and evaluate a national control program. But the other added aspect of our research group being involved in this is really this incredibly valuable position that we found ourselves in, because we are researchers by definition. So this has given us an opportunity to conduct some basic scientific research through a national control program. So how are we going about with this monitoring and evaluation? We have two resources at our disposal. The first one is that of the 3 million children that we're treating in Zimbabwe, treatment is being given at the schools. So that's the only place you can get treated during the mass drug administration program. It runs for a week. So basically everything stops in Zimbabwe for a week. All the schools, 4,000 of them, administer praziquantel. Each child who's given praziquantel, they're recorded in that national MDA register. We record their name, their height, their weight, because that's how we give the drugs, the drug dosage, sex, and every time they come up, we have that. So for the next 5 years, each child will get entered into the register. The second thing is we can't check every one of the 3 million children involved. So we've got 20 sentinel sites in Zimbabwe. So these are dotted all around the country, and we do very intense sampling and sample collection in those 20 sites. The 20 sites are randomly distributed around the country to capture differences in infection transmission, ethnic groups. We know that in Zimbabwe there are two main ethnic groups, so we don't know if there are any genetic differences in the pharmacodynamics of the drug, especially when you give it so repeatedly. So that to us gives us a resource of 5-year data from 4,000 schools and 3 million children. So every year we're collecting this data from these children. What sort of data are we collecting from our sentinel sites? We're looking at the efficacy, so infection levels and morbidity. Is the drug actually effective? We're looking at short-term effects, so the adverse events and immune reactivity. We're looking at the longer-term effects on the host health, so the hygiene hypothesis plays into here, and the long-term effects on the parasite population structure, which, as I said, I'm not going to dwell on today. The first result, this is a graph of our M&E activities from the 2012 and 2013 drug administration program. So every year, those 3 million children are going to receive praziquantel for the next 5 years. So we look here, if we look at the white bars, this was the situation before we started treatment, and you can see about 80%. And those of you who are familiar with a lot of our immunology work, this is Murewa, where we conduct a lot of the immunology work we've published and we've talked about a lot. If we look at the green bars, that's infection level after the mass drug administration in 2012. So we have a very strict time period of when we have to go and sample. Then we went back in 2013, which are the black bars, and we treated again. And then we look at the post-treatment levels. So that's 2013. 2014, we are awaiting the mass drug administration. Our team in Zimbabwe is already out doing the pre-MDA sample collection and looking at the parasite levels. Those of you who are very observant will have noticed these 4 groups where the white bars are basically the same as the green bar. So this is 2012 pre-treatment and post-treatment. Those 4 areas were deliberately left in 2012 and not treated to see if there's any issues with drug performance or anything of that nature. So that's why the white bar and the green bar are the same, because we sampled them at the start of the MDA program and then 6 weeks later, which is the prescribed time for sampling, and you can see there's no change. When they were recruited into the treatment programs in 2013, you can see that dramatic decrease in infection levels. So what we can confidently say is that the mass drug administration program in Zimbabwe has effectively reduced schistosome prevalence in the country. Okay, I'm going to move on to the immune responses now. Now, I mentioned earlier that the reason really for giving this talk was to try and show you the basic scientific research that underpins a lot of the advice we give to the stakeholders and the policymakers. And we're focusing on immune responses because in people who are exposed to schistosomiasis, I mentioned that you do get the development of protective immunity. So for 2 reasons, we're very interested in looking at immune responses because of the development of protective natural immunity. People get infected when they become in contact with infective water, so whether it's domestic chores or recently new domestic chores or just having fun. So they're going to be continually exposed. So we're interested in the development of protective immunity, but also in vaccine research, because it is going to be very difficult to eliminate schistosomiasis and thus achieve the recent World Health Assembly resolution for elimination of schistosomiasis purely on drug administration. So I'm going to show you some immunology, which I promise not to dwell on too much. What I'm trying to show you here is the level of detail we go into when we do our immune studies. So here, for example, we're just looking at the specific antibodies. Antibodies are the effectors of the immune responses. And what we have here is just a proteomic analysis of parasite proteins. So we take basically ground up parasite proteins. We run them on a gel, first by charge and then by weight. And then we take serum from people who are resistant to infection. We use it to screen the parasite proteins, and we see which ones they react against. This is a very good way for looking for vaccines, but also for us to demonstrate what people are responding against. So what we can see here is schistosomes do induce a very strong immune response in the antibodies, and the antibodies are important because those are the effectors of the immune system when it comes to schistosome infection. We've also then decided, well, what is the effect of treatment on parasite responses? The way praziquantel works is it goes into the body and it binds onto the surface of the worms, gets transported through calcium channels, and then causes muscle paralysis in the worms. Once it causes the muscle paralysis, the worms can't contract and they can't move so easily, so the surface tears. When the surface tears, it exposes a whole lot of proteins to the immune system and induces immune responses. So actually, for my PhD, this was one of the very first things I published because it wasn't known before, so it was very, very exciting. And so I was interested to see if treating people basically boosts the immune system. Does it vaccinate them? And what we see here is the first thing it does is I've got two antibodies here. The blue one is an IgA response and the purple one is an IgG1 response. We know that the IgG1 response is protective, and we know that when we look with people who are residents of schistosome endemic areas who never get reinfected, they have very high levels of this IgG1 response but very low IgA. So I was curious to see if you treat these people, do you then induce an IgG1 response and shut down the IgA response? And that's exactly what happens here. So our treatment here really is acting like a vaccine. We can prove it further by looking at those immune responses. Before treatment, people are recognizing a certain amount of parasite proteins, but after treatment, they recognize more. And what's really special about this graph is that all these proteins that I have highlighted in red, those are the schistosome vaccine candidates. And this one here is Biovax, which is now in phase 3 clinical trials for schistosome treatment. So what it tells us is that praziquantel alters antibody responses. We can go further. This is really to show you that we've been busy in my lab. We've looked at the antibody responses. These are the cellular responses. So from all those people who are exposed and people who are protected against infection, we have collected their white blood cells and been doing a lot of analyses. We've looked at their B cells, myeloid-derived dendritic cells. We've looked at their T cell subsets. We've looked at their monocytes. We've looked at their dendritic cells. We've just phenotyped them, and we've done functional assays, and we've been very busy, and we've published quite a lot of that work. Why am I bothering showing you this? Because the immune system requires a very intact T cell response. And I've picked just one of the results to highlight the innate lymphoid cells. These cells are very important for directing the immune response either towards pathology or towards resistance to reinfection. They are very recently described, so described in the past 2 years. And the reason I'm very excited to show you this result is because we're the first group to show that they do exist in human populations from an African background. This has never been shown before. And secondly, that they are altered by helminth infection. So as I said, the work is submitted. And I've put this here in case there are some hardcore immunologists who really want to know how we classify those cells, because you have to use very specific markers to show what they are. But that's not really the purpose of what I'm trying to show you. What I'm trying to show you here is when you look at people who are infected, so they're egg positive, positive for the parasite eggs, their proportions of those cells are significantly lower. And as I mentioned earlier, you need those cells in order to direct your immune response towards either pathology or protection. That difference is really important in the youngest people who are being infected for the first time. Now, two questions you can ask here. Are these people egg positive because they started off with low levels of those cells? So were they going around with low levels of those cells and that made them susceptible to infection? Or did they have normal levels of infection and of these cells, and then they were infected and that dampened down their cell levels? Now, if the second is true, we should be able to restore the levels of these cells back to this level if we treat the people. So that's what we did. And isn't it nice when it works? Here we go, 6 weeks later, we treat people and the levels of those cells have been significantly increased. And if we look at people who were negative before treatment, and we now compare the levels of people who were positive before treatment, but are now negative after treatment, what we show is we've brought up the levels of those cells and we've restored them. So that's really an important aspect of our studies. So what I can say here is praziquantel restores the ILC2 proportions in people who were infected. We go on further. We've also looked at cytokine responses. Cytokines are mediators of immune responses. So basically, they connect various aspects of your immune system, particularly between your cells that can sense the parasite and the cells and antibodies that can deal with the parasite. So very, very important. A lot of people will focus on a single cytokine. We don't do that in my research group for 2 reasons. Firstly, because although people can move from one state and end up with pathology or end up resistant, the way they make that journey is different. So they may use different pathways. So if we focus on a single cytokine, we're going to miss a lot of the heterogeneity that is going on here. The second thing is cytokines don't act individually. There is a lot of interaction and cross-regulation. If we focus on only one, we could miss a very important pathway. So we thought, go for the lot. So we've picked up various cytokines that are associated with different immune phenotypes. So innate, T helper 1, T helper 2, and T helper 17. And they all do different things, and they participate to varying extents in pathology or immunity. So we've looked at all these. And I've extracted just one figure. As you can see, this is D and E. So there's A to C missing from here, but I didn't want to drown you in immunology. And what we have here are cytokine responses, so interleukin 17, interleukin 21, 23, and interleukin 10, that are specific, so they're induced in specific response to the infective stage of the parasite, CAP, the adult worms, or the egg stage, which causes pathology. And what I just want to draw your attention here is that when we treat people, we move some of them from different levels of cytokines. So some of them go down and some of them go up. So this is really very important result showing that praziquantel changes the cytokine profile. Now, that's all very exciting. It keeps the immunologists in the lab very busy. What does it mean to you when you're a host on the ground? Is it of any consequence? Is it just the dynamics of the immune system responding to what's going on in infection? What we want to try and do is see what the health consequences are for the people who exposed every day. And we've gone further in our work just published last year. What we've done is we know that when you treat people, some of them remain susceptible to infection and they become reinfected. Some of them become resistant. So we followed these people for 3 years, and so who remained uninfected, so these are the red ones, and who became reinfected, which are they were blue when I made this slide, they are now gray. So the gray ones are the people who become reinfected. So we want to define a phenotype that is associated with resistance and one that is associated with susceptibility to reinfection. This, coupled with experimental studies, show that this particular phenotype that we induce is actually protective. So to the person who's doing their day-to-day domestic chores, the important aspect here is that praziquantel treatment reduces reinfection rates. We have given the mechanisms of how it does that, but the readout that is important to the population is that you can reduce reinfection rates by treating people. I've gone further with that, and actually, I think last year that we published this, suggested this is actually a very good model for an immunization. You can use infection treatment as a model for vaccination. We do it in the veterinary field, things like theileria, caldriosis. You give an infection, you treat, and that vaccinates them against subsequent infections. So this is effectively what we're doing here. People are already infected, we treat them, that boosts or accelerates the immune response, and that reduces their reinfection rates. I am not alone in saying this. 2 other groups that have been working extensively in Kenya have also proposed this as a paradigm for vaccine development for schistosomiasis. What about the effects on other parasites? I mentioned earlier, and I made a point of highlighting the little chap with hepatosplenomegaly, that you get co-endemicity between Plasmodium and schistosomes. So what happens in a situation where you've got a person that is infected with schistosomiasis and Plasmodium? What would happen if you then treated the person for schistosomes? Well, two things can happen. You can make the malaria worse, or you can make it better, or what we prefer is it does nothing. Okay, so what we did here is we looked at Plasmodium-specific responses in people who were infected with schistosomes that we treated. So here we've got results for the people in blue were people who were not treated, and the people in red are the people who were treated for schistosomiasis. We did nothing to the Plasmodium. First thing we did was we just looked at whole Plasmodium responses, schizoned. And what we see is no change, so treatment has no effect. The next thing we went into was to look at specific antigens, and we chose the MSP1 and MSP2 antigens. We chose those for two reasons. The first one is a scientific reason, which is that MSP1 and MSP2 are vaccine candidates. So what you don't want to do is to go into a population, treat them for schistosomes, and make them unresponsive to any potential vaccine candidates. Because when the Gates Foundation or somebody else comes with their MSP1 vaccine and they vaccinate those people and it doesn't work, you're not going to be very popular. So we checked that what happens when they are treated, what's the MSP responses. The other reason we chose the MSP1 and 2 candidates is because we like to keep our friends happy. And the MSP1 and MSP2 antigens were discovered by a colleague of ours in the same institute, David Kavanagh. So we like David, so we thought we'd use his antigens. The other thing we did is we checked the immune responses against schistosomes. I've told you that we alter the immune responses. So just to be sure that our experiment really was working, we have our internal control, that the immune responses against schistose should shift. If these ones don't shift at all, then maybe our drugs didn't work or something like that. This one here, what we actually do is we look at immune responses after treatment and we subtract them from pre-treatment, and that's why this is negative for something that actually increases. So this experiment is really nice because you get the changes you expect against schistosome antibodies, and you don't get anything against malaria. So this is really nice because what it says is that praziquantel does not affect Plasmodium-specific immune responses. Okay, again, that's nice if you're being academic, but what about if you're the person on the ground? Well, what we also checked was the incidence of malaria and the severity of malaria, so clinical reports of malaria in the areas, and what we get is no increase in malaria incidence and no change in malaria severity. So this is good, it's looking good. Okay, now we have to address the hygiene hypothesis. First proposed by Strachan in 1958, sorry, in the 70s, looking at children born in 1958, so he followed 17,000 siblings. And what he suggested was that a lack of early childhood exposure to infectious agents, microorganisms, and parasites increases susceptibility to allergic disease by suppressing natural development of the immune system. So basically what he means here is dirty kids are healthy kids. That's the gist of the hygiene hypothesis. Now, I have taken this figure from a meeting of clinical allergists held in Zimbabwe, I think in 2011. And this is their understanding of the hygiene hypothesis, that as the prevalence of infections increases, you decrease the prevalence of allergy. Now, what we're doing is the opposite. We're starting here with high levels of helminth infections, and we're pushing the kids that way. So this would predict that clinical allergy should go up. So we decided not to shy away from this and have a look. And we addressed this through a series of studies. So here we're looking at schistosome infection. Here this is Nadine in our group who was looking at allergy and autoimmune reactivity. So what are the questions we're asking? Is there a relationship between schistosome infection and bystander antigens? Bystander antigens refers to all the other antigens, things like allergens or autoantigens that are around in a person that's infected by schistosomes. The second question we're asking is, what is the effect of praziquantel treatment on the bystander antigens? We measured this in two ways. We looked at antinuclear antibody ELISAs. So antinuclear antigens are your nuclear proteins. When you have autoimmune diseases or certain autoimmune diseases, what has happened is that your immune system has started reacting against those and destroying basically your cells. When you go into a clinician's office and say, I think I have an autoimmune disease, they will do a series of tests. The very first one they will do is an ANA ELISA. And they use this for looking at different reactivities to different antigens. And these are an initial diagnostic step for your different autoimmune diseases, such as systemic lupus and other diseases. The other thing we did was to look at allergens. So we did this by a skin prick test. We looked at various allergens. I've just put the 4 major ones here. House dust mite, cockroach. I didn't know that German cockroaches were different from African cockroaches, but apparently they are, so we had to use both. And grass mix and aspergillus. This was informed by actually a lot of data from our allergy colleagues in Zimbabwe. And what we eventually discovered was that the most common allergic response in Zimbabwe is to the house dust mite. So we then went on further and did immunoassays using the house dust mite allergen called DERP1. So what do we find? If we look at anti-nuclear antibodies, so these are anti-nuclear antibody levels. This is schistosome infection. As you see, people with very few schistosomes or no schistosomes have very high levels of anti-nuclear antibodies on average, and people with high schistosome counts, people like 800 to 1,000 eggs per 10 mils of urine, very low levels of anti-nuclear antibodies. The blue line represents what your clinician would be looking at. This is the cutoff point. So if you came in and had levels above this blue line, then they would do further investigations if you didn't have any other reactivities. These ones here, they are not considered something to worry about. So what we can see is a negative association. If you have very high levels of schistosome infection, you have very low levels of anti-nuclear antibodies. We also looked at the skin prick reactivity. So here, again, you see this is levels of schistosome infection, and this is skin prick size. So you'd go in and you'd get your skin prick, and then the size of the wheal relative to the positive or the negative would say whether or not you're atopic by that description. This graph looks a bit odd because in that graph we have corrected for the host age and host sex, which would also affect the size of your wheal. So that's why it looks like we've plotted the residuals. So taken together, what this say is that yes, there is an association. It's a negative association between anti-nuclear reactivity and schistosome infections, and as well as atopic reactivity to house dust mite. Okay, so what happens when you treat? If we look at the anti-nuclear activities, levels go up significantly 6 weeks after treatment. So we get a significant increase in ANA reactivity 6 months post-treatment. If we look at atopic reactivity, so we've done this by ELISA. We've looked at 2 villages. This is a low schistosome infection village, so we didn't really expect much to be going on here, which is not surprising. And this is high schistosome infection area, so you get the very high levels. And as usual, we have our internal control of what happens to the schistosome immune responses. The reason I'm looking at IgE and IgG4 is because IgE is the antibody that mediates a lot of the allergic reactivity, but it is modulated by IgG4. So if you have high IgG4, you can modulate the activities of IgE. IgE is also an important response associated with protection against schistosomes. So it is nice that we get our anti-worm IgE going up, but we don't get any increase in the IgE directed against the DERP1, the house dust mite. And in fact, in this low infection village, it goes down. So what we conclude from that is that there's no significant increase in atopic reactivity 6 months post-treatment. So I come back to the question that the local person in the street will ask. Is this relevant to me? What is the clinical significance of those findings? And I was really pleased we went to have a look at this, because talking to the clinicians in Africa, where we've had control programs, they are not reporting increases in allergic cases or in autoimmune cases. So what on earth is happening? So I put this figure again, but at this meeting that was held, as I say, with a whole lot of African clinicians, this is the picture that they are reporting from the clinical data. So what is going on? At this stage, I should say, I heaved a sigh of relief because I did not want 3 million children in Zimbabwe to be coming down with allergies and autoimmune disease and have my name on all the papers that say, go and treat them. So this was very good. So we asked what's going on, and in discussions with a colleague of mine in Zimbabwe, he is an allergy clinician and autoimmune disease clinician. He is the only one in the country, so he runs the only reference immune disorder clinic in Zimbabwe, and more importantly, he also runs the reference clinic for the SADC region. So he is very knowledgeable and a very reliable source of data. I said to him, what on earth is going on? And we've come to sort of think what's going on is you have your allergens there, they cause an immune response, and a whole lot of exciting things happen, and then that leads to your clinical symptoms. So what we are seeing clearly is people are being exposed to the allergens. When we give them the allergen through the skin prick, we can measure the levels of these IgE antibodies that are present, and we can also show the reactivity on the skin. But it's not translating to the asthma, the eczema, hay fever, or anaphylaxis. And what we think is happening is somewhere along here, you have a homeostatic mechanism that is stopping the immunology from translating into a clinical outcome. So that's the reason that you're not getting this translation. And I was really pleased to see some of the work presented at the food allergy and safety assessment workshop that's just ended in August in Kenya, where a lot of allergy clinicians said exactly that you have a lot of allergic reactivity, but it's not translating to clinical symptoms. So that is good, but we want to be sure. So we thought in our research group, we better investigate this a bit further. The way we've gone about it is to look at mechanistic studies in the different immune disorders. We have access to patients who are suffering from immune disorders. So all along up until now, I have taken you from patients who suffer from schistosomiasis, and we're looking at the allergic reactivity or autoimmune reactivity. What I'm doing here is going from the other direction. These people have clinical symptoms. They have been diagnosed as having allergy or autoimmune diseases. Let's have a look at what's happening in their immune system. So we've got about 70 people from the clinic in Harare, and their clinical symptoms have all been confirmed following the international guidelines for how to confirm different autoimmune diseases and allergies. So we've had some serological assays conducted in Zimbabwe, and we're conducting some immunoassays and some molecular assays. I just want to give you a snippet of data. This is really hot off the press. You can't really see the labels here, but we've got different types. We've got food allergy, we've got inhalant allergy, we've got atopic and autoimmune represented by those different bars. And what I just really want to highlight is we've looked at the cytokines, and you get differences, things like interferon gamma popping up in some conditions but not others. We've also looked, and this is very important, IgM against schistosome adult worms, diagnostic for schistosome infection. These people have all seen schistosomes, and yet they've got all those allergic reactivities. And some of them are even beginning to develop immunity against schistosomiasis because they've got IgE. So this is very important that these people basically have the comorbidities, which is exactly what the clinicians mentioned at that meeting in Harare in 2011. That's enough of hygiene hypothesis. This is the last aspect of the work that we've been doing that I want to talk about. I've showed you what's happening at the short-term level. Now let's have a look at what happens at the long-term level. For this, we've taken a mathematical modeling approach. We're using pattern-oriented modeling. Now, we're very excited about this. At the time, I was working with some super clever mathematicians, mathematical biologists, and we empirically derived a method for modeling our immune responses. and then predicting immune responses and transmission and infection levels 5, 10, 20 years down the line. This is very useful because you can then have your intervention and see what would happen. We've published the method in PLOS Computational Biology and some of the predictions in PNAS, but we were very pleased with ourselves because we'd come up with a very exciting mathematical knowledge, and it was a novel way of doing this, and when we were writing up our work, we discovered that we were reinventing the wheel. Somebody had published that method in Science in 2005. Would you believe, in connection with thinning of forests? So it's not surprising we hadn't seen it before, but it was quite nice that we found it ourselves, actually, and not somebody else had to come and tell us about it. So if you read our PLOS Comp paper, you will find the reference to thinning of trees in that methodology. Anyway, what does it do? Basically, they're models of parasite-antibody interactions. They capture population-level field data patterns. So a lot of the patterns we see with the immune responses, they capture that. And once we've confirmed that our models are really reproducing what we see in the field, we can then trust them enough to go and predict what will happen in a couple of years' time. Just in case there's some mathematicians who really want to see the modeling framework, I have put it here. And basically what our model does is it captures the infection process, what's happening to the worms in red, and it captures the host immune response in blue. We capture that in 2 sets of differential equations. Then we solve these differential equations having put in values for all the different parameter estimates. And again, if you're really that excited, go and read the PLOS Comp paper. Okay, so what does it do? Well, what we find is good for us because it confirms our immunology, but also a little bit worrying. Here we have our antibody levels. One is at baseline. So what we're going to do is look at the change in immune response relative to where the person started. And then look what happens over a 15-year period. And we've looked in 3 different villages with 3 different infection levels. So high infection area, a medium, and a low, because this is what you find in the field. And the WHO recommends that if you have low, medium, and high, you treat, and you treat at very specific intervals and with specific frequency. So we've taken all that into account. And what happens is you go in here, you treat. So the stars represent the 5 years of treatment. And this is very important because a lot of the national control programs are running for 5 years. So we've got our 5 years of treatment, and what happens the first year is you boost the immune system. This is good. This is what all our immune studies show. And then as you go on, you treat, and the immune system is still high relative to where you started off. And this is because the more frequent you treat, people are carrying fewer worms, and therefore the boosting is a lot less. So there we have it. After 5 years, you stop treating, because that's when the responses, the MDA programs run. And what happens is that the immune responses fall, and they fall to below the levels that you started off with. What are the consequences of infection? Well, what you have here is, here we're looking at egg count, so relative to baseline. So you start off there, and you have lower levels of infection. I've shown you this because I've shown you that the MDAs are very effective at reducing infection levels. Then you stop treating, and people start getting reinfected. They get reinfected to levels higher than they were before treatment. Now, this is not novel to schistosomiasis. Some of you will have heard of all the controversies of impregnated bed nets for control of malaria. In young children, if you give them impregnated bed nets, you don't expose them to Plasmodium infection. Then when they're about 10, 11, or 12, and you stop using the impregnated bed nets, the kids get bitten by infected mosquitoes for the first time. They have no natural immunity to the parasites, and they have more severe malaria at that age than they would have had they got what we call premonition, initial malaria infection. So this is what is happening here. And that's very important, because the message we can then take to the politicians and the policy holders is that schistosome intervention requires sustained control efforts. But more importantly, also that relying on just praziquantel treatment is not going to do it. If you're intending to stop in 5 years' time, you have to be thinking ahead. What about providing safe water and sanitation? What about vaccines? What about vector control? That's very, very important. So I can summarize what I've presented today and show what the effects of praziquantel are. First of all, it's efficacious. This is good. It induces and accelerates the development of protective immunity. This is also very good. It does not affect Plasmodium responses or incidence of malaria. That's also good. It may affect allergic and ANA reactivity, but this has no clinical consequences for the people. And really, really important, the message we really want to give to stakeholders, governments, anybody interested in control is that it's got to be sustained. If it's not sustained, it could actually result in reinfection overshoot, which takes the people to a worse off position than they were before they started. I shall stop there and thank all the people in my research group here in Edinburgh who've been involved in this work over the past sort of 4 or 5 years. Got several collaborators here in the UK, Mike Turner, who's now gone off to the Wellcome Trust and left us to run our gels by ourselves, and Richard Birchmore, who does the proteomics. Colleagues in France who do all the cellular work for us, and colleagues in Spain. Particularly want to thank my colleagues in Zimbabwe, Taka and Nicholas, because the two of them and myself and our research groups formed the Understanding Bilharzia program. You'll find a lot of our results and a lot of the work that we do under this banner, and also Elo Pisibanda, who runs the allergy clinic. Of course, none of this work would be possible without our participants in Zimbabwe. If they didn't take part in the studies, we'd have no study participants, and our funders. And finally, I'll thank you for your attention.